Today we will discuss about a new topic, that is how the calreticulin can migrate on the cell surface on the melanocyte apoptosis. So far we have understood that calreticulin is a ER soluble protein and it is present within the endoplasmic reticulum. However, this paper discusses about the translocation of calreticulin on the cell surface of the melanocytes, and that can also help in the activation of the immune response. So what's the hypothesis behind this paper? We will discuss that thing in this video, and in the consecutive or subsequent videos we will discuss what are the experimental they have done and how they have validated their hypothesis. So this calreticulin is a ER soluble protein. It is usually found in the endoplasmic reticulum, that can be seen here. However, when the ER stress is when the oxidative stress occurs, it will trigger melanocytic ER stress, and that will cause unfolded protein response as and that will unfolded protein response and that will leads to the overcrowding of the endoplasmic reticulum with the unfolded protein, and that will trigger one of the three unfolding protein response, that is PERK pathway, IRE1 alpha and ATF6. And among this PERK is found to cause this translocation of this CRT from the endoplasmic reticulum to the cell surface. And this CRT, that is here the calreticulin is represented as CRT, and this CRT then helps in the activation of the immune cells such as dendritic cells, activation of these dendritic cells. Along with that, the recruitment of these CD8 plus T cells and the proliferation of the CD8 T cells towards these migrations to the site of infection, and thereby it helps. This process helps in taking care of this main reason for this oxidative stress. And in this figure, the stress is caused due to this reactive oxygen species on the epidermis, and the UPR response that has occurred because of this kind of stress is this overproduction of this HSPA5, unfolded HSPA5 within the endoplasmic reticulum, and this overproduction of this HSPA5 will then disrupt this PERK pathways, and will then disrupts this PERK pathways that prevents the activations of this PERK, that is PERK EIF2 alpha phosphorylations. So this PERK EIF2 alpha phosphorylations then trigger the translocation of this calreticulin, which is the ER soluble proteins, to migrate to the cell surface, and the immune response that I have just discussed in brief will occur. However, they have not mentioned too much about the role of the other signaling pathways that are also triggered because of the endoplasmic reticulum, that is IRE1 alpha and ATF6. And we know that the role of this ATF6 is to cleaved off and activate the proteases in the Golgi apparatus, and simultaneously they will also cause this, all signaling pathways will cause the epigenetic changes that will lead to the production of proteins that are essential towards controlling this ER stress. At the same time, it will suppress the production of proteins that are not essential at the present conditions. And we will discuss subsequently how they have designed their experiments and what are the results that they have developed or they have got, based on which they substantiate their hypothesis. So if you need to understand more about this paper in detail so that you can understand the subsequent videos better, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.